A drug can be defined as any substance used for the purpose of diagnosis, prevention, relief or cure of a disease in man or animal. Drug development. Drug development is a term is used to define the entire process of bringing a new drug or a device to the market. It includes drug discovery development, preclinical research, and clinical trials, and few people still refer to the drug development as more preclinical development. Prodrug concept. A prodrug is a pharmacological substance administered in an inactive or significantly less active form. When one administered, the prodrug is a metabolized in vivo process termed as bioactivation. A prodrug is an inactive derivative that will be converted into the active drug in vivo, and it is a precursor of a drug, and it must undergo chemical conversion by metabolic process before becoming an active pharmacological agent. Soft drug concept. These are opposite of prodrugs. These compounds are designed or synthesized as active compounds that readily undergo metabolic inactivation to non-toxic products. Types of drug design. There are two major types of drug design. First one, ligand-based drug design, structure-based drug design. Ligand-based drug design. Ligand-based drug design or indirect drug design relies on knowledge of other molecules that bind to the biological target on interest. These other molecules may be used to derive a pharmacophore model which defines the minimum necessary structural characteristics of a molecule must possess in order to bind the target. In other words, a model of the biological target may be built based on the knowledge of what binds, and this model in turn may be used to design new molecular entities that interact with the target. Alternatively, a quantitative structure-activity relationship in which a correlation between calculating properties of molecules and their experimentally determined biological activity may be derived. This quantitative structure-activity relationship in turn may be used to predict the activity of new analogues. Structure-based drug design. Structure-based drug design or direct drug design relies on knowledge of the three-dimensional structure of the biological target obtained through methods such as X-ray crystallography or NMR spectroscopy. If an experimental structure of a target is not available, it may be possible to create a homology model of the target based on the experimental structure of a relator protein. Using the structure of the biological target, candidate drugs that are predicted to bind with high affinity and selectivity to the target may be designed using interactive graphics and the intuition of a medicinal chemist. Alternatively, various automated computational procedures may be used to suggest new drug candidates. Pharmacodynamics. Pharmacodynamics, which may be defined as what the drug does to the body. It comes from the Greek words pharmakon, meaning drug, and dynamikos, meaning power. It is the study of the biochemical and physiological effects of drugs on the body or on microorganisms or parasites within the or on the body and the mechanisms of drug action. The relationship between drug concentration and effect. One dominant example is drug receptor interactions as modeled by ligand plus receptor It gives the reaction dynamics and can be studied mathematically through tools such as free energy maps. Pharmacodynamics is often summarized as the study of what the drug does to the body, whereas pharmacodynamics is sometimes abbreviated as PD. Pharmacokinetics. Pharmacokinetics may be defined as what the body does to the drug. It is abbreviated as PK. It is from ancient Greek words. Pharmakon means drug and kinetics means to do with motion. It is a branch of pharmacology dedicated to the determination of the fate of substance administered externally to a living organisms. In practice, this discipline is applied mainly to drug substances through in principle concerns itself with all manner of compounds otherwise delivered externally to an organism such as nutrients, metabolites, hormones, tonins, etc. Pharmacokinetics is often studied in conjugation with pharmacodynamics. Pharmacokinetics includes the study of the mechanism of absorption and distribution of an administered drug. The rate at which a large actions begin, the duration of the defect, the chemical changes of the substances in the body. Example, enzymes. Induced fit theory. It is applied to drug receptor interaction. The induced fit theory has been based on the hypothesis for which recent evidence is being accumulated of induced conformational changes in enzymes. The hypothesis has been developed by a number of authors. For example, Koshland postulated that the active site of an isolated crystalline enzyme does not necessarily need to for morphology that is complementary to that of the suitable as a kind of negative to it, but that it acquires such morphology only after interacting with the substrate, which induces such a conformational change. He postulated that the active set of the enzyme is flexible or better plastic or elastic and not rigid, that is, not only can it be deformed or altered, but it also possesses the ability to return of the original form after being deformed. According to the induced fit theory, the biological effect produced by drugs arises due to the activation or deactivation of enzymes or even of non-catalysis proteins through a reversible perturbation or change in tertiary structure enzymes or proteins. Conformational change does not get restricted, however, to protein. Drugs having flexible structure can also undergo conformational change as they approach the site of action or the receptor. Drug receptor interaction therefore can be seen as a dynamic and in most cases reversible topographical and electronic compromise or accommodation between drug and receptor stimulates which gives rise to the biological effect. Recently, Koshland and co-workers have modified the induced fit theory so as to explain cooperative effects, the phenomenon that binding of one ligand molecule somehow accelerates binding of subsequent ones. Hence, binding of the first ligand to a polymeric protein, which may be an enzyme containing a receptor, induces a conformational changes in one of its subunits. A change in the shape of this subunit influences the stability of the remaining subunit. The resulting energy of stabilization makes possible stronger binding of the next molecules. Occupancy theory. This theory was correlated by Clark and Gordon. This theory, also called template theory, states that the intensity of pharmacological effect has been directly proportional to the number of receptors occupied by the drug. According to this theory, drug-receptor interaction which comply with the law of mass action may be put by the equation R plus D equilibrium or D, where R represents a receptor, D is molecule of the drug, RD the drug receptor complex, E is the pharmacological effect, and K1 and K2 the rate constants of absorption and desorption. The number of occupied receptor proceeds has been found to depend on the concentration of the drug in the compartment of the receptor and on the total number of receptor in the unity of area of volume. The drug effect gets much more intense as the number of occupied receptor increases. Gets maximum action is corresponding to the occupation of all receptors. Rate theory. On the basis of the postulate of Craig-Sato and Haldobro that a drug is efficient only at the moment of encounter with its receptor, Paffon and co-workers gave a rate theory. According to Paffon, activation of receptors has been proportional not to the number of occupied receptors but to the total number of encounters of the drug with its receptor per unit time. Unlike previous theories, the rate theory does not need formation of a stable. Structure-activity relationship. The development of physical models for drug action using essentially property-based parameters reflecting changes in hydrophobic, electronic or steric character. Property-based parameters can be criticized, however it is not always easy to measure or calculate such parameters. Particularly, steric effects and the properties which are likely to be relevant. Further, more property values often are reductant, that is, two or more structures can have the same pKa values, and after all the drug designing structure is formed. In sharp contrast to a QSAR model built up from a relationship between activity and physical property, in which the structure of a molecule is concordant. Basically, the structure is a count of some primitive components or features. Quantitative structural activity relationship. Classical QSAR methods describes structure activity relationship in terms of physiochemical parameters and steric properties methods. Especially comparative molecular field analysis consider the 3D structures and the bindings modes protein ligands. Quantitative similarity activity relationship desire for relation between the similarities of individual compound under biological activities. Theory and methodology of these approaches of describe together to them the proper use of particle least squares analysis for deriving quantitative structure activity relationship. Drug receptor interaction. Structurally, non-specific drugs action results from their physiochemical properties, and structurally specific drugs action results from their chemical structure. Drug action is due to drugs bindings to the receptor site. Drug binding results with ionic or non-ionic binding. Non-ionic binding is due to hydrogen bond binding, hydrophobic binding and van der Waals forces. Receptor binding. The binding of ligands to receptors is governed by the law of mass action, which relates to the large scale status to the rate of numerous molecular processes. The equilibrium dissociation constant is defined as ligand plus receptor gives LR, where L is ligand or receptor. In one way, to consider the effect of a drug in which the response is related to the fraction of bound receptors is known as occupancy. The relationship between occupancy and pharmacological response is usually non-linear. This explains so-called receptor reserve phenomenon, that is, the concentration producing 50% occupancy is typically higher than the concentration producing 50% maximum response.